Taking the standard approach of having these 1.5%, it's predicted that we need increase per year, is there any opportunity to give a substantial increase in yield? We may not be able to do it quickly, but can we do it at all? And it was recognised by a lot of scientists for a long time that C4 plants with a more advanced photosynthetic system, tend to outperform C3 plants by about a factor of 50%. 3 would be nice, of about 50%. And that's shown in this inset here, which shows a range of different C3 data for a range of different C3 and C4 crops that have different lengths of growing season. And you can see that C4, no matter what the growing season length was, tended to outperform their C3 partners by about 50%. Now, a 50% increase in yield is a huge increase, and wouldn't it be nice to put that into rice? And this is just showing the picture in the background is some data from rice grown at IRRI, just a small trial experiment. And we have a C4 maize crop in the background yielding about 13, 14 tonnes. And rice giving 8 to 9 tonnes. And that too represents about a 50% increase in yield. So could we ever convert rice into this nice C4 crop with this increased potential? So the difference between a C4 photosynthetic pathway and a C3 photosynthetic pathway is shown in this schema here. C3 photosynthesis all occurs within a single cell. And within that cell, carbon dioxide is taken up from the atmosphere, fixed into carbohydrate, and then it's that feeds the plant. And that's a very standard pathway. The main catalyst for this is rubisco. It's a carboxylase that fixes the carbon dioxide. And unfortunately, it was designed a long, long time ago when the geological CO2 concentrations were much higher than today. As the CO2 concentrations have been depleted by the success of the vegetative biomass on the planet, the efficiency of this enzyme has gone down and down. And at current levels, about 30% of its activity is spent not fixing carbon dioxide, but fixing oxygen. And that leads to loss of carbon dioxide and a waste of resource and a waste of photons that were fixed by the electron transport chain. In the C4 photosynthetic pathway, they have developed a two-cell anatomy. One cell is like a carburetor or a turbocharger that collects the carbon dioxide from the atmosphere, delivers it to a bundle sheath cell where the photosynthetic apparatus is, and releases that carbon dioxide. The system is improved because this carboxylase here, PEP carboxylase, is a very efficient CO2 scavenging enzyme, and it doesn't undertake any oxygenation reactions. So it can work at very low CO2 concentrations. That means then that when it's delivered into the bundle sheath now, if the bundle sheath is impermeable to carbon dioxide, the carbon dioxide in here can accumulate to much higher levels than current atmospheric concentrations. We're currently about 400 parts per million, and we think the concentration inside the bundle sheath may well exceed 1,500 parts per million CO2. So then that allows rubisco to function much more efficiently, as if it was back in geological time, as it were, and we have much higher rates of photosynthesis. We also don't do any wasteful oxygenase reactions, and so we don't waste any of the sunlight, or we waste far less of the sunlight. And so this is actually an improvement of light use efficiency. So this means then that on a square meter basis or a square hectare basis, there are more photons available to make carbohydrate than there are in a C3 plant. And how does it achieve that? Well, it has to achieve that by altering its leaf anatomy. And this is a typical rice leaf here, where the veins are far apart and we have just a bag contained by the epidermis of mesophyll cells that are all independently fixing carbon dioxide, delivering them to the veins, and then that's distributed. Within a C4 plant, it's slightly different. The Bundle sheath cells have become much larger, in some species anyway, but these now contain a lot of chloroplasts and do the photosynthesis that they can deliver directly to the veins. And then there's a single layer of mesophyll cells surrounding that bundle sheath that act as the turbocharged CO2 pump, pumping carbon dioxide into these photosynthetic cells. And so there's a definite order, if you like, of vein with a bundle sheath, a mesophyll, then it's got its other bundle sheath and a vein. So it's a very ordered anatomy compared to its C3 counterpart. And the benefit is shown in this graph here, which shows that actually this is a CO2 on the CO2 concentration on the x-axis, this is photosynthesis on the y, that you can see that C4 plants, sorghum and Soteria shown here, are able to photosynthesize at much lower concentrations of CO2 than C3 plants. So we have an improvement of light use efficiency, we have an improvement of photosynthesis, and there are a couple of other benefits too. One is that because C4 plants can fix and do good rates of photosynthesis at very low CO2 concentrations, they can close their stomata on the surface of the leaf. And because they can close their stomata, or close their stomata more than a C3 plant, they lose less water. And so a C4 plant also is much more water use efficient, probably double the efficiency of a C3 plant. The other gain is in terms of nitrogen. Because C3 plants have an inefficient rubisco, they make massive amounts of this single protein. And this protein takes up a lot of nitrogen. As soon as you go to a C4 plant, the amount of protein that's required in the leaf is far less. And so you get also an improved in nitrogen use efficiency. Now these are genuine increases in efficiency. So how do you go about doing that? Well, the first thing that you've got to do is you've got to start developing the biochemistry so that you have the appropriate pathways expressed in the appropriate cells. You need to have bundle sheath and mesophyll cell-specific expression of certain enzymes. We have to reduce the activity of certain enzymes in cells. So, for example, the photorespiratory pathway will have to be removed from rice, or at least reduced. But then there are a load of other structural things that have to go alongside this. For example, to get really good diffusive communication between the mesophyll and bundle sheath, you have to have lots of plasmodesmata. And the way that you regulate plasmodesmata at the moment is largely unknown. The bundle sheath size and plastid number within those bundle sheaths has to be optimized and increased. Vein spacing has to be checked. And then the bundle sheath cells of C4 plants are highly suberized to make them largely gas-time. And the genes that regulate suberin production and their deposition in specific cells, again, is unknown. So there's loads of unknowns. So this is just a summary of what I've said, and I've got a lot of slides, so I'll skip past that one. But I think it was generally recognized that the C4 photosynthetic pathway was one of the few evolutionary mechanisms that we know about that could actually feasibly enhance photosynthesis and therefore yield in some sort of substantial way. Another interesting fact is that C4 evolved relatively recently, over the last 35 million years, but it's done that independently 62 times in very many diverse species. C4 monocots, on the other hand, and dicots that are differentiated by about 180 million years ago, and yet both dicots and monocots have evolved C4 photosynthesis, and they all have evolved it via the same underlying mechanism, which is really interesting. There's only one solution to the problem, and that's happened many, many times. Another interesting factor is all the genes that we know of that are involved in C4 are also found in C3 plants. And that gives us hope then that we can find simple regulatory switches that might turn these genes on and allow us to get a C3 rice converted into a C4 much more easily than you would think, looking at the enormity of the problem. So I put that cheeky question there. It can't be that difficult. So our basic philosophy at the moment within the C4 consortium is C3 evolved to C4. There were some anatomical changes, some biochemical changes, and then a bit of fine-tuning, and we end up with C4 photosynthesis. Should be easy. 3 years ago, the C4 Rice Consortium was funded. It was set up about 10 years ago. It took them 7 years to get money. But 3 years ago, it was funded initially by the Bill and Melinda Gates Foundation, but now with other donors as well. And that involves a whole range of scientists around the globe that have got together to try and solve this problem. The timeline, well, when the donors are around, and I hope they're not in here, we say 15 years, but probably 25 years in reality by the time we're able, or we think we'll be able, to put C4 rice-like plants out in the fields of farmers. Phase 1, the one that we're in at the moment, we're just starting the second 3-year funding period, and that started in May this year. And that's all been about gene discovery. What are the genes that regulate these different processes that I've mentioned so far? How using that information, can we switch those particular traits on? And so this sort of summarizes our basic approach. We're using a mutagenesis approach. We're looking to activate genes in rice. We're looking to knock a C3 plant. Can we activate a gene in rice and make it more C4-like? Can we take a C4 plant like sorghum and mutate it and knock some of that that C4 function down? So that's being done. We're using bioinformatics to screen transcriptomes to see if we can, I'll tell you a bit more about that in a moment, to see if we can use bioinformatics to come up with genes that are different. And we're screening genomes and transcriptomes of C3 and C4 lineages, so if we can see what the basic differences are between these plants. That candidate gene list then is used to produce transgenic lines for testing the function of these genes, and we then need detailed physiological analysis to see what the effects of those genes actually have had on photosynthesis. So I'll just tell you about some of the teams that we've got set up. We've got a molecular engineering team. These are scientists you'll probably recognise: Julian Hibbard and Jane Langdale from the UK, Peter Westhoff from Germany. And so one of the things that's happening is in the consortium labs, like Julian's, they're making the constructs that we need. Construct production is actually quite a slow process. I think it was as Kiefer mentioned earlier, and that is a limitation to the rate at which some of the work progresses. They're sent over to IRRI, we make the transgenics and check we've got the appropriate expression levels. This is our progress to date. All the genes in green, we have successful transgenic lines that have the appropriate mode expression level. I might add that getting the sorts of expression level that you need for a C4 plant is really difficult because those levels are exceptionally high, and the only way that we found of doing it is by using full-length promoters from C4 plants. We've got some other genes that we're doing at the moment, and no doubt the bioinformatics guys will tell us others that we need to find to do. And then we're pyramiding these lines to stack them, and at the moment this is our first line that we've stacked. We've got 3 genes into the same line. 2 plants look rather similar. They don't seem to have any huge phenotypes at the moment. And so that work is ongoing. The Langdale lab is looking at very early leaf development, just when the vascular bundles and the veins are developing, to look to see if we can find C4 specific genes that are being expressed at these very, very early stages, just as vein deposition is occurring. And they've come up with really nice candidate lists now, and they're going into our transgenic program for testing. And I can't tell you any more about those because that's where we are. We're developing promoters all the time to give us appropriate cell-specific expression, and that's been done in the Westhoff lab. And this is an interesting one here, looking at 4 different PEP carboxylase, that's the carboxylase that we want to transfer over, looking at the promoter region of that particular gene in 4 different species of C4 from the Panacoideae. And we can see that they all have quantitatively give very high GUS expression when expressed in rice. And that expression is very cell-specific, specific to the mesophyll cells, even though it's moving into a C3 rice species. So this has been quite a surprise to us. This is the control here for 35 GUS expression, showing that that's expressed everywhere. So we really are just getting mesophyll cell expression in these particular lines. So we're trying to develop new promoters all the time to help our process. The Brucknell lab are looking at the developmental gradient you get along a monocot leaf to try and find out which of the genes are being switched on at different stages. And they're using transcriptomics and RNA-seq to do that. And they've come up with some fantastic profiles that were recently published in their plant cell paper. I'm not going to tell you any more about that, but they've given us lovely candidate sets of genes now that we will test both in C3 and C4 plants. We have a bioinformatics and systems biology team. They've been working on a range of projects. One of them is the GAIN-1 1000 planters transcriptome project, and we have 39 C4 and 26 C3 plants within that project. And this group now is analysing the data that has just recently been made available from those. I've missed the pipeline. To cut a long story short, they're seeing genes that are C4 and C3 specific. There are large numbers, but those with high significance are shown here. 300 that are up in C4 and 200 that are down in C4 or up in C3. And we're going through the bioinformatics of those genes now to see if we can identify novel candidates that we want to take forward. If we look at those genes on a map pathway scheme, what we find is all the appropriate genes are up and down regulated as you would expect within that gene set. So we're hopeful that there are other genes that we don't know about in those gene sets that will be able to help us. We've got a mathematical modelling group led by Xinwang Zhu, and this is really important for us, because we really need to know what is the precise architecture of the metabolic pathways that we put into a C4 rice and whether it will function. And in silico testing is so much faster than anything that we can do in the lab. So we're optimistic, but it's an extremely difficult challenge. It's not just coming up with a metabolic pathway now, we've got a 2-cellular metabolic pathway that has diffusional interactions that require both concentration gradients to drive movement and also catalysts, enzymes that act as transporters that facilitate diffusion. And this model is now largely complete and testing is underway at the moment. I'll skip that one. The other interesting factor is that because we have this diffusional model, we have to now consider things like the diffusional gradients of gases, of water, of carbon dioxide, and I missed out on the previous slide of light penetration. And he's making really good advances in the models that allow us to predict metabolic pathways in a spatial 3D scale, and I think this is really exciting. We have a molecular physiology team and a genetic screening team. A lot of these people are in the audience, and you'll hear talks during the week about various aspects of the work I'm summarising today. At IRRI, as I mentioned earlier, we're doing the mutagenesis screens, and so we need really simple high-throughput screens. These chambers here just show you some that we've built that are able to screen plants at low carbon dioxide concentrations. I'm not going to be telling you much about those today, but it's got to be easy if we're going to screen large numbers of lines. And so one of the processes that I told you was we screen rice activation tag lines, and we've screened about 60,000 of those now, either through populations that have been sent to IRRI or through us going to Sumei and Jinan's lab in Taiwan and Korea to screen their lines in the field. And we've screened 70,000 sorghum lines for a reversion of the C4 pathway. One of the simple screens that we've been looking at, and that's what I'll finish off telling you about, is vein spacing. This is an easy way of assessing whether your bundle sheath cells here have fewer mesophyll cells behind them. Remember, this is what we're trying to achieve, this stoichiometry here. You can see veins with a little handheld microscope. And so it's very easy in the field to spot a conversion like this. We've been doing that. This is again saying that you can use CO2 concentration to screen as well. If we grow plants at a concentration where this arrow is at 20 ppm, C4 plants survive, C3 plants die. So you've got a life-death method of picking, choosing whether you've got an appropriate mutation, even though it's a complex biochemical trait. So this is some of the lines that we've got from the activation tag. And what we're showing now, this is a wild-type rice line with about 9 mesophyll cells. We're getting veins that are much closer together now in some of these mutants. Sometimes they're smaller cells, sometimes they're fewer cells, and these are quite stable, and we're taking those through to analysis at the moment. The phenotypes of these plants are varied. Sometimes the phenotypes are associated with a dwarf-like phenomena, and sometimes they're not. So we're hoping that we'll be able to find a vein spacing trait that's just a vein spacing trait rather than being associated with other morphologies. We've been screening sorghum. We've stopped doing that now. It's such a big and slow plant. We don't like it anymore, and we've switched to Ceteria. But we have screened those. We do that in high throughput screens. We grow about 70,000 plants a week, and we can screen about 24,000 of those. And we're getting vein density mutants in those. This is a typical mutant with many cells here, 7 cells between the veins, when it should only have 2. And you can see from this top picture here, this is what you see through a hand microscope, where the veins are much wider spaced. So we're trying to identify that gene. The way that we're doing this is we're looking for allele frequency of the causal SNPs that have occurred during that mutagenesis program. And the way we're doing it is to backcross and clean up that line. At the moment, we've sequenced plants from the BC1F2 population. We get a nice segregation showing a single gene Mendelian inheritance. We have 8 mutants from those 70,000 lines, which is not many, and only one of them have we got through to that stage where we are doing sequencing at the moment. The rest are in progress. Blue squares means in progress. Some preliminary data, this came into me this weekend, was our strategy was to take 45 individuals with a 35 times coverage for sequencing from the mutant and the non-mutant pool, and then to screen those for variants that showed separation only between mutant and non-mutant pools. So this allowed us to get rid of a lot of the background noise, and this followed the method of Zhu et al. In Genetics, published in 2012. We found about 500 variants. 50 of these were significant. 40 of these are non-synonymous, so could be an amino acid change. 25% are synonymous, and 35% are intergenic, so probably not so interesting. And we're hopeful now, we're going through these manually, sufficiently low numbers, going through these manually to use literature and database searches to find interesting candidates. So that was my final slide, my conclusions. Well, we think the C4 Consortium has done this the right way. We've set up a global consortium of people. We think that's necessary, and I think several speakers have mentioned that this morning. We're using high throughput sequencing to sequencing both the transcriptomes and genomes to try and give us a faster handle to isolate the genes. I think as Aero mentioned this morning, speed is imperative. It's very easy to let 3-year grants just go by. Our screens are being successful. We've got some anatomical screens, and I haven't told you about, but we've got some compensation point screens as well. And so we're optimistic that using a sequencing approach we'll be able to get to those genes reasonably quickly. Of course, with the taglines, it's much faster. And then many of our biochemical genes have now been transformed into rice in a cell-specific way at an appropriate level. And finally, acknowledgements. This guy here, John Sheehy, in the middle here, he was if you like, the father of this project. He was the one that got off his butt and got all those global scientists together. He recently retired and I've taken over. And I'm not the head of the project. I like to call myself the coordinator, because it's coordinating global activities that we need. So I'd like to thank the IRRI team. There are a large number of people working at IRRI, very, very hard indeed, and the C4 Rice Consortium in general. I'd like to thank Bill and Melinda Gates for funding us. Also, the British government has put in a large chunk for this phase 2 of the programme. And if I can encourage any of you guys to encourage your governments to put funding into this, it doesn't have to come to us, it can come to you, because all we need to do to be faster is to increase our capacity. Thank you.